So yeah, so he's the faculty at the Department of Physics at the University of Toronto, and he did his PhD training at Princeton University and also the postdoc training at UC Santa Barbara, and he's very interested in about the cell-cell interaction and tumor heterogeneity, and sounds very cool. And also, he got a lot of nice publications on them in the high-impact journals, so very impressive. So and let's welcome him to join our workshop and give a wonderful talk today. Yeah, thank you.
Thank you for inviting. Sorry, we can't hear you quite yet.
Okay, maybe it should be on now. So I'm going to talk a little bit today about sort of, I don't have a lot of immune connection, but I think it's still about relapse. So I think we're trying to go towards immune, but I think this was a good setup for us to get there. So hopefully you guys will find it interesting. So I wanted to sort of just lay out a very caricaturish view of tumor dynamics and what parts I'll talk more about. I mean, the idea is that, of course, we generate tumors all the time, and thanks to our immune system, kind of keep it in check, right? Many of our tumors can be sort of very slowly growing, which I'm going to call equilibrium phase, so it's not exponentially growing. Tyler had this nice plot of linear growing tumor, which he also called the sort of equilibrium phase. So it's sort of something like that, where you have a little bit turnover, but it's not just exploding on you, right? And then the worst part happens where it kind of, I'm calling it the escape phase, if you will. And escaping, I think, is really from the immune system in some ways, right? You can kind of hide from it or figure out a right microenvironment. There are many, many variables to think about how it escapes, but there's a phase like that where it kind of grows now. And this is the phase where people think sort of a lot of heterogeneity develops, right? Partly sometimes there is sort of a mutator syndrome in the sense that you don't have a good way of error correcting and whatnot, right? And so the idea is that why tumors are hard in some ways is that you have a very heterogeneous population. You can target some. Of course, we learned yesterday and in many talks, there are fixed mutations. They are like BRCA2, there are many mutations which are going to be everywhere. But the challenge always has been like, oh, this is a mass of a lot of things going on. How do we tackle it? And they're all interacting, right, with each other in many ways, positive and negative interactions, and with the immune system, right? So there's a lot. It's a very interacting system, right? And I mean, I'm preaching to the choir, but is a good place for people who can think about modeling and statistics and quantitative might be useful, right? If it was nice logic of few genes in and out, you don't need that much mathematics to go beyond it, right? So I think this kind of makes it ripe for what we can try to do. So today, what I wanted to talk about is two projects. One sort of thinking about how does this tumor heterogeneity change and respond to when you have drugs. So you give it a drug, how does it respond from there? How does it inform relapse? How does it go down? What does this heterogeneity do to it? And most of the work I'll talk about will be in these two papers, which are last several years now. The other project, which is more theoretical, because early phase stuff is hard in the sense that, well, what do you measure? You always know about cancer here. Although there are some now cohorts where people have been, for example, there's this big cohort in UK where they have been sampling blood forever. And then many of them get tumors. So you have all this pre-data for them. So there are various groups thinking about it. But we haven't, but we wanted to think a little bit more theoretically in the sense, what is the tumor initiation? And there's this big idea that there's a microenvironment feedback, right? Can we think about that in a conceptual way, and does it tell us something which might be interesting? And we're starting to work on this sort of, I'm going to call ecological dynamics of this interacting cell population using actually some of the kind of data Trevor was showing yesterday, or through this cell-free DNA, right? Because you need something. or cell-free or whatever temporal dynamics, right? Once you have temporal dynamics, you have some chance of kind of getting behind these interactions. I won't have too much to say here, but I'll be happy to chat with people if they want to. So let me start with this. This was work in collaboration with Catherine O'Brien, who's a pathologist here at Princeton's Margaret and UHN. And Sumi was a postdoc. She did all the experiments mostly, and Sophie was a physics graduate student in my group. So what is the problem? The problem we wanted to ask is, well, you have a very heterogeneous population, let's say, to start with. You give it drugs. Well, most times the tumor actually regresses, unless you're just giving a wrong drug. The mechanisms could be many, many different ones. You know, you're doing immunotherapy, you could be doing chemotherapy, radiation therapy. I'm not thinking about that at this stage right now. So the tumor regresses. And you have what people call this minimal residual disease and whatnot. So there's something has happening where everything looks good. And you need to stop treatment. You know, the tumor is gone, you don't want side effects. And that's where the chance of relapse is there, right? Of course, there's a chance things just coast along, and that's amazing, as we saw in some of the patients Trevor had yesterday, that, you know, they just flatline, which is amazing, but things do relapse, right? They could relapse from the source site, they could relapse from a metastatic site, many ways that can happen. And the hypothesis had been, or still there, I think, is that, well, the relapsed tumor is definitely less heterogeneous, right? Because something out of whatever was there, not everything could survive when you give drugs. Some of them are sort of more likely to survive because of their genotype, right? You have this huge genetic heterogeneity, something survives, and then those are the ones which live. So in principle, if you can understand what the nature of these things is, you have a chance of doing something more. Okay? So that's the underlying hypothesis. And the other thing is, you know, what's happening here. That would be pretty amazing to understand more too. So those were the two things we wanted to understand in this project. And the way we sort of, or Catherine's group approached it really is in a xenograph model, right? And to track sort of this heterogeneity in time, if you will, they barcoded those cells. So these are genetic barcodes. So there's a little piece of DNA inserted almost randomly into the genome. This one is actually, I don't think, too random. It has a very site which is kind of, it's a hot site where it goes in, but the barcode has variability, right? So some barcoding schemes are done differently, where you put the same piece of DNA, but where it integrates tell you, differentiates cell to cell. This one is actually having the sequence which is different. So you take those things, put it on the back of the mice, and then depending on whatever you want to test, you can do treatment, you can do no treatment, etc. And then in the end, you basically take these tumors out and you sequence them. And the sequencing is basically telling you what is the composition of the tumor. Is that good, right? So the composition is basically how many barcodes, how many colors of these different barcodes are around. Yeah.
So these barcodes are inherited?
Yes, they're genetic.
Okay. So the barcodes are inherited by division.
Yes. So they're genetic barcodes. They go into the DNA and they just get passed on.
Yeah, just to clarify the experimental technique, but how does it tell you something if it does about the other genetic material in the specific tumor cell? So like, how does it help?
No, this is just a tracking. This is just able to track. So I have, let's say, 20,000 cells I put into the mice. Each cell has a color now. So now when I look at a later time, I can say how many cells came from a single cell at a later time.
But do you know the properties of these cells or something about them?
So good. So these cells are from colorectal cancer. This
no, I mean, like, from the point of view of heterogeneity. Like, you know, which
genetic heterogeneity.
Yeah, you know, like the barcodes in the cells, they're different.
Correct.
Okay, but like, how does it help you to know the properties of the cells?
Very good. So again, to set up the question, right? So one is the genetic heterogeneity in the problem, and we sequence that, and you find there's a lot of heterogeneity. What we wanted to really track is how individual cells are contributing in time. So you might have two cells, which have the same genotype, but they'll get different colors in this.
How do you match genotype and colors?
You don't match. You just put color right now. So the worst case would have been the whole tumor was homogeneous, but you still have different colors on them. Does that make sense? Yeah? See?
I guess from that perspective, if it was actually homogeneous, you would expect like the frequency not to change probably over time. Like you
Yes,
can
exactly.
still
So
infer back some.
precisely. So we're going to think about those things. Exactly, right? Good. Is the setup clear? Yeah. So what happens when you just, on the mean field level, the tumor size and whatnot. So this is a slightly busy plot. Don't have a pointer, but I see this hook. So this is the tumor volume, which is on the back of the mouse, right? And this is just time. These are the different treatments, so to say. So you let the tumor grow for 5 days or so, and then the flank of the mouse, and then at this arrow you put treatment. Treatment might be saline, which is like a control, just salt water. DMSO is another kind of control for some drugs which need sort of hydrophobic stuff to dissolve in. 5-FU is another drug, CPT11, Folfari. These are Folfox, another one, I don't know why it's missing, but there's all these different chemotherapy drugs, right, which were given. Of course, it depends on the dose and whatnot. In this experiment, for CPT11 and Folfari, you see basically regression. So the tumor doesn't grow until you take the treatment out. So you take the treatment out, and this thing then comes back up. These guys had showed no response to the treatment, at least the doses we were using at the time. Good. So what's the idea again? Well, the starting population had lots of colors, right? So this is nice. I mean, nothing died in principle. Of course, there's going to be some biases in grafting and whatnot, but you don't expect a lot of change in diversity, or who's going to show up at the end of this tumor. Good. This one we expect, much less diversity. So you can go and measure it. And to clarify that this is not a resistant tumor. Yeah.
Oh, sorry. So in the green line there, is that do you have like sensor data, or are those actual measurements?
These are actual measurements.
Okay, so that's okay.
This is like 10 mouse or something like that.
Right.
Oh yeah, 9. So this is green is for 9 mice. This is the size of the tumor in the back of the flanks.
Okay.
That's it.
Thanks.
Yeah.
Just a very quick question about the mice. Are those immunodeficient?
Yes, these are nude mice, and this is a human cell line. Absolutely. Sorry, I should have mentioned that. Good. So we are not expecting a lot of immune response, but I mean, I think there's some innate response which people think about and stuff like that. So good. So our questions were around these things, and this is not a resistant. So you can actually take this tumor, put it back. If you give the drug, the tumor doesn't grow. If you give DMSO, it grows back. Right? So it's not a resistant tumor, it's a relapse. Good. Relapse could be driven by resistance, but this is not. Right? It's an important part of what we want to study. Of course, if you so we did some control where you can let the drug on, and then you will see resistant tumors come up, and they would not show any more sensitivity to that drug. Right? So that works too in this system. So what did we want to do? We really wanted to quantify all this sort of composition, right? So you can go and sequence them, right? There's a target, you know exactly what you want to sequence. So it's cheap, as Trevor was saying, you're not doing anything else. You just go and PCR that part up and you sequence that. This is going to become a little bit easier as we go. It's a slightly busy plot. Each dot here is actually the size of the number of cells. So this is the T0 sample. This is the sample we put in, right? So each one is kind of equally available. That's what this is supposed to show. It's going to become a little easier as we go on. So what happens when you put salt? Well, these are different tumors. And now you can see some of the clones are massive, right? The scale is, this clone might actually be almost 90% of all the tumor in that mice. Right? So this is sort of almost, this blob is 60%. So you can see some of the clones become massive, right? And the others remain small. So what do we expect? And again, but it's not the same clone. It's a different clone in each run. Right? The T0 sample was the same. You just see different clones in different mice. I'm calling, so I just want to make clear, I'm calling it a clone, but this may already be a genetically heterogeneous population. Because while growing, mutations might have arose, right? For me, the definition of the clone is it came from the same cell. Good, so that's the definition we are running with. Well, these were the other ones where tumors just grew. Drugs didn't really do anything. So again, maybe not that surprising that some different clones become big and small, right? So it's basically like there's no drug. The drug didn't do anything. There's no selection. The idea of drug is it's selecting, right? It's killing something specific and something special remains, right? What surprised us is that pretty much the same thing happens when during relapse, right? So things which regress. So this is sequenced after relapse. Right? So you have enough cells that will come up, and you can't really tell the difference between this and this if you just barely look at it. Right? We're going to go into a little bit more quantitative way of thinking about it. So that was the first surprise, right? That our hypothesis was we're going to find some special clones here, and then we can go dig back into the genetics of them and figure out how we're going to sort of, at least in this mouse model, think about a genetic basis of relapse. So I think we took two things out of it. One is, well, I'm going to plot some distributions soon. The big clones are massive, but the variation is really large. You know, you actually see some 100,000-fold difference between the biggest and the smallest clone, right? Which kind of speaks to strong selection, right? Something is getting selected. I mean, one clone really became 80% of all the tumor. And I'm still postulating there's no selection, or at least that's what it looks like. But the surprising part is there's no correlation between dominant clones from tumor to tumor. Right? That's what tells you that perhaps selection may not be an important. Yeah.
Just look naively at this picture, asking kids, what the order here? It looks very wonderful.
Yes, completely.
What that means?
That we'll try and answer that, right? So that was precisely the surprise. And this is where Catherine thought it might be useful to talk to talk to people who it's just not obvious what to do with it, really. You know, and then what theorists do, the only contribution theorists have to the world is plotting the data, right? And we plotted the data, and the way we plotted it was make a cumulative distribution, right? So this T0 is the cumulative distribution of what you started with. You still see variation. Right? Some clones are still bigger. This was just growing them in a dish, right? So you take this clone, you grow them in the dish. But what is surprising is across all the other treatments, this is the distribution you see.
Sorry to interrupt you.
No, please.
Question from probability
Yeah.
side. I'm a probabilist. What does it mean? Distributions are identical, and then there is a variability. Do you mean that they belong to the same class of distributions?
Well, I have
Or
just
are
plotted
they
it.
exactly
I
identical?
have just plotted it.
Huh?
I have just plotted them, right? You can tell me if they look similar or not.
Somewhat, yeah. I see subtle difference.
Yes, no, there are, you know, things like that. Absolutely, absolutely.
And that's why you have
Quotation marks, right? How do you want to think about how identical?
close, yeah, let's see.
Yeah. How
I had a question. So these are fractions of the total,
these are so you can plot the fractions, right?
but the total number changes.
The total number, well, depends on the size of the tumor,
Uh-huh.
right?
So,
So the total
so the
number
fra-
scales with the size of the tumor, if that makes sense. No.
right, but if you look at the raw count,
Yeah.
right, what do you see any differences?
Not really.
Uh-huh.
Not really.
Okay.
I mean, you might see that in some tumors. So for example, it really depends on how much you sequence, sadly, right? So there's a big variable of how much you sequence. Rather, it's not even tumor size. If you sequence more, you see more counts. You sequence less, you see less counts. So I don't think we saturated the diversity in any which way. If we sequence more, we're going to keep sampling the bigger clones, I'm sure. We'll also keep sampling smaller clones, if that helps you, right? So there are even more smaller clones. So this was already a million reads per tumor, right? So your smallest clone is 10 to the minus 6, right? Or 2 times 10 to the minus 6 or something like that. So if you did 10 million reads, you'll again find even smaller clones. Good. So, well, so in terms of really defining what this distribution might be, what we did was, well, we can kind of fit these distributions. This is a classic gamma distribution. And this is the cumulative form of gamma distribution. I'm sorry, this is some incomplete gamma function. You mathematicians. All right, okay, I don't have to apologize. Sorry? Yeah, right? So K is just the size, and these are the 2 parameters. Shape parameters, exactly, right? And the other one is basically in this part, almost like a parallel, right? But yeah, exactly, right? So log linear, there are many names for this thing, I guess, right? So you can, so what I meant by identical is you can fit this slope in some maximum likelihood way, and you kind of get numbers between 0.3 and 0.5. I have a plot later if you want to see it. That's why I sort of pooled them together. So the distribution is basically this. So these are cumulatives. So this distribution, sort of for the bulk of it, looks like this. And this one is your classic gamma distribution. Good. So these are the 2 distributions we see. And so this is what I mean by quantifying the randomness, right? So it is pretty random, but yet there is some sort of pattern in the randomness or in the chaos, right? So the question for us was, well, the original hypothesis is gone. We don't know exactly if there are no special clones, yet we're kind of claiming that you get these really dominant clones somehow, and we kind of have to get them from some model which doesn't invoke selection, right? So I thought I'll spend a couple of slides thinking about this clonal dynamics, and then we can get back to what we learned biologically from this. Oh, by the way, this is, yeah, I thought people might enjoy. So this is sort of different tumors. Remember, each one was coming, each line there comes from 9 or 10 distributions, 9 or 10 tumors. So this is how I was drawing these solid lines in the previous plot. This is how the variability looks around it, which is kind of crazy. It's like, I mean, admittedly, this is log scale, but you got to choose the resolution at which you want to see things, right? That's a choice you make. And at that resolution, I mean, this is kind of crazy that different mice kind of behave. There's a little bit differences, but within that pool, it's very tight. So what we wanted to ask is, well, how can the simple theory question was, how can such large heterogeneity sort of emerge? Right? How do you get these sort of big and small clones, this kind of things, in a very simple way without having any selection, if you will? So so I'll just run through very simple models, I guess, just to give some intuition. The simplest version is you have some exponentially growing thing, right? You have a bunch of cells, they grow exponentially. Let's say they have the same growth rate, right? Coming back to making some null model for the problem, and then we can think about why it doesn't explain the data. Here you get, so each clone is just exponentially growing, and your clone size distribution turns out to be exponential. Right? If you have a simply exponentially growing population, the clone size distribution is completely exponential. Right? And the mean is sort of whatever the size of the mean size is there. Scale depends on when you're looking at it, right? Good. Now you can say, well, maybe tumors are a little bit more complicated than this. Maybe what you put in are some sort of progenitor-like cells. They transition into something, and then it grows. There could be multiple stages into it, but at least put in one stage and see what it does. And in this model, immediately gives you a gamma distribution. So this is what we were already seeing. So this is what we saw in the dish. Right? We saw exactly a gamma distribution in the dish. Right? So this is not a terrible start for explaining at least the phenomenological properties of this tumor cell line in a dish. Right? I know we have lots of clinicians, so I want to be careful in not claiming more than that. Yeah. Yeah. two mics.
Anyway, yeah, I understand that there is probably a reason why you write mu is like 1 minus mu is.
Yeah. I'm going to come to that.
Minus 1. I understand you probably have reasons
Right.
for that. Is there like a reason for that? And also, would you have like mu between 0 and 1?
Yeah, so in this model, yeah, good. So good, very good. So here in this simple model, I mean, the tumor is only going to survive. There's also a term. So here, this nu is sort of a ratio of theta and gamma, right? So depending on what the rate of input and the growth is. So imagine theta is very small, right? So you have once in a while you transition into this thing, and then this thing explodes. Then you get sort of a slightly heavier tail of the distribution, right? That's where this nu is going to be small, and you get like 1 over k type distribution, right? You still have sort of lots of stuff, bigger clones, because whoever got first into the room got to just explode, right? When theta is small. When theta is larger than lambda, if you will, then lots of clones are entering all the time, and you kind of get more like a Gaussian distribution, right? When theta becomes bigger than 1, right? So this is the exponential cutoff, and then this thing becomes rising. So, right? So you kind of basically get, so rather than getting these kind of shapes, you start getting these kind of shapes. Good. So that's just the ratio of these two parameters, theta and lambda. Good. But okay. But the shape we are after is this, 1 plus alpha. You can't get that from this, right? This model will not give you this. Doesn't matter what the parameters are. Good. And we want to show there are various models where selection will give you things like these, but we were kind of in a bind that we want to get something like this without any selection, right? So you can get this quite easily, even starting from something like this, if you allow these growth rates to be different. Right? If these growth rates are different, you can get any distribution you like. You can just put that in the distribution of the growth rates, and you get whatever you like. Is that makes sense, right? So in some ways, sir? Yeah. Okay. I mean, this is the problem with lots of us who do stuff. You get to a problem and you don't read anything and you just start doing stuff. But yeah, I'm not claiming this is new. I mean, I'm sure somebody did it like that. So coming back. So remember, there's no selection, right? So we want to write sort of, I'm going to use this word neutral dynamics or equipotent dynamics, and we wanted to write down something like that. So what turns out, I think, there's a simple intuition, right? How do you get selection when there is no selection, right? You want to reduce competition somehow, right? And if you have spatial structure, you can do that, right? So the simple intuition is you can still have your one state model. You have some hierarchy of progenitors and rapidly cycling cells, but now there's spatial structure, and the bigger clones really are not competing with each other in some ways, right? So they can get even bigger, if you will, right? So you can think of a rough intuition as you can think of sort of a colony growing in a dish, right? So let's say these yellow cells entered. So first cell which entered was a red cell, made a big clone. After a while, a yellow cell entered, make a bigger clone, but this size of this clone is not affected by the red cell, right? If you have a room of a fixed size, whoever gets first fills it up. People coming after are, jostling for places, right? So that competition is now damped down. So now many clones should be able to become big, right? So that was the intuition we were going with, and queen cell. So what this looks like is the growth is now not exponential, but it sort of has an interaction term, if you will, right? So for example, a colony growing, a 2D colony, alpha is a half, right? So the growth is only on the edge, for example. So 3D colony, so it's kind of depends on the dimension. Of course, tumors are not just 2D or 3D balls. They have their own structure. So, alpha is your favorite number. The data tells us what alpha is. I'll come back to it in this minute. So here alpha is some term which tells you how sort of spatially structured the tissue is. Again, trying to be very, very, the word I'm going to use is like phenomenological, right? So just the simplest parameterization we can do to keep making progress. And of course, there's some very detailed models one can make of the spatial structure. But for now, I'm just putting it as one parameter. So again, the intuition is, if I'm tracking number of these rapidly cycling cells, a red cell might come in, start growing, and at some, characteristic time scale, a new cell will come in, could be yellow or red. Let's say it's a green, but kind of dies off quickly. It doesn't really grow that much, and a yellow cell. So that's the kind of stochastic dynamics that's going on. And if you really wanted to compute the distribution at some time t, what you're interested in, what are the sizes of clones at some time t, right? And not all the clones might survive, by the way, right? So in those distributions which look nearly identical, not all of them have the same clones. That's clear, right? Lots of them has gone to 0 in one, they're present in the other. A different clone is big in this and small in that. That was very clear. But even the clones that have died are quite different, right? So, but the distribution is something you can compute. So you can do this calculation, in full-blown glory. But the simple intuition is, sort of there's a Poisson sort of waiting time for stuff to come in, right? And if it comes at some time, sorry, this should be t prime. This should be t prime. Sorry, this should be t prime. So nothing comes in until t prime. Something comes in, and then this clone grows. And if it gets to size k, that's the probability of seeing a clone of size k at time t. Yeah, so exponential wait time. Yes, yes, yeah. It's a Poisson process with exponential waiting time. Correct. Thank you. Right. So good. So this is the. So even this very simple way of looking at it, even if you ignore the birth death stochasticity in these clones, you can actually get the intuition of why you would get the kind of distributions you want. So now what is happening to each clone? It's not growing exponentially. It's suppressed by the mean, right? The size of the total tumor.
I have a question. I mean, I'm a little bit not sure if I'm on the same wavelength here, but do you actually want to use some kind of like ecological niche theory to describe this kind of thing with your limited competition from spatial structure? But like, how can you actually find any argument for that in the context of a solid tumor? Like,
there's tons of niches going on here, right? I have no access to them. I can write down more. I can write down a model with more parameters.
Like this. You have this like hypothesis about this thing, but like, does it have any like
basis? Oh yeah, for sure, for sure. So people have sort of tracked. So what you can do is you can do these even in not you forget tumors. You can take like skin and color a few of the stem cells. And you will see that there's a clone of different colors happening at different places, right? So you see this structure very, very easily, even in a normal tissue, not to talk about tumors, right? So there's this great work by Ben Simons at a slew of work, and he and his colleagues did, where they were tracking just in normal tissues, these sort of, clone size distributions. And for many of those cases, they found sort of near exponential distribution in the stem cell population. I'm not looking at later ones, and then did some stuff. So does that help? Sorry?
Just a small comment. This phenomenon underlies that, like the Kelly-Coquet's effect, they have 3 colors and they can be only females. So that's basically because of the X and Y chromosome effect in the
sure.
Oh, like something that can like. Yeah,
I wasn't going for that, but we have 1,000 colors, right? But yes. But
so yeah, I have a comment and a question. So the as a biologist, I think like very simple explanation for that question is you have the spatial issue. So it means that if you have the cell near the blood vessel, you get more nutrition, and you're far away or in some dead area without nutrition, and then it grows less.
Right.
So that's the Hamburg rendering. But my question being, it keeps in my head about that. It's the So for example, you touch about the spatial structure issue. So you have a large solid tumor and you treat with drug, and the outside will usually get killed compared to the tumor inside. So do you think that those become less colorful, become oligoclonal? Sometimes is that just because by chance, by survivability?
It would have, if the number of colors went down, you would have gone down that path. But we're seeing the opposite. The problem we had is colors are not changing. We basically see the same kind of representation of the tumor in the relapse, even when they were sort of dead for a long time.
My question is, like, maybe it's not a surprise after all. So, like, let's imagine you have 1 billion of cells. They're different. Like, say they're 100 clones. Yeah. Then you like administer the drug, but not in the like killing off those, and you're remaining with like minimal residual disease, but it's still, it can be like 100,000 of cells.
Absolutely.
So it's still like a large sample enough to for all the
So sampling wise, yeah,
to fit
absolutely.
you. While you like continue doing the therapy or like increase the dose, you may be left with like 50 cells. Oh, and then that's like you cannot choose 100 of them.
So the surprise is not that, it's not the surprise that we had enough cells, so we should have enough clones. The surprise is, why is the drug not killing some things more than the other? Does that make sense? Why are some genotypes less and more sensitive to the drug? Because that's our picture, right? When you administer a drug, the idea is that the clones which come back are somehow able to evade the drug, right? So those genotypes are important, right? Some mutations are important because they sort of give it ability, at least that was the hypothesis.
Yeah, but well, it should also depend like on the phenotype, on the cell state.
Oh, absolutely.
The stochastic process, which would explain like under moderate dose, yeah, it should not, the resistance should not be prominent, while under like the prolonged treatment or like very big dose, yes.
I mean, so in this case, at least the two populations we had, one where the dose was low enough that they keep growing. So we were hoping at least we'll see some difference between the tumors that kept growing and the tumors where you could actually not detect any, drug response where you could actually see no tumor on the back of the mice. The cell's there for sure. I'm going to come to that. We want to understand those cells. So the next part is going to be about that, right? But it's not that we left some giant tumor there. But what this is saying is exactly what you said. Those cells are still around. And they don't care which genotype they come from. Right? That's what we are learning, right? I mean, a bit of this math is a bit of a digression, which I thought you might enjoy, just to figure out how do you get these distributions, right? And so I had some fun with it, basically. So, you can have these clone sizes now, which have sort of a very different growth rate. Not exponential, but they're controlled by the rest of the stuff which is in the pool, right? It's trying to grow exponentially, it's getting suppressed by everybody else. And this is sort of the mean field for the total tumor size, right? Which is basically this equation, but now I'm also allowing stuff to come in. So you can just put in mean field dynamics here, and you basically immediately get sort of this distribution, right? And you can calculate it in the full sort of, you can write down the full stochastic version of this and it gives you, but this kind of tells you that this equation alone is basically gets this alpha shows up here, right? The same alpha which is controlling the growth here shows up there.
What would be typical values of alpha for your experimental data?
We measured it, right?
It can be any number, but
No. So this alpha we got from the data.
yeah. But what would be typical values produced by the data? Typical range.
So I think.
Oh yeah.
No. We can go back. There's no harm in that. So these are the ranges in which we are seeing this value.
So alpha is like 0.3, 0.5.
Yeah. Right? Because it wasn't clear that this alpha is going to be the same alpha there, right? And that was sort of the theory helped us, that we actually learned something, a very phenomenological parameter about these tumors. And it's not surprising that if you do different cell lines, by the way, I didn't get to it, a different cell line or a different primary tumor will show a different set of slopes, right? So this number depends on what you're putting in. This number also depends on how many cells you're putting in. So something which so if you actually put in very few cells, they don't crowd up. They start giving you gamma-like distributions, right? So we then tested all of these ideas to make sure that we're not fooling ourselves, right? But okay, so this is sort of just this idea that you can get these things quite if you kind of invoke this and you learn this parameter directly from actually learning the distribution. There's only one parameter to be learned, so I didn't want to put more parameters in my model than I could learn from that distribution. Okay, so just to summarize, you can now do, depending on whatever the alpha is, you get different slopes. You can do simulations. You can do all those kind of things. Stochastic simulations kind of works out. So this led us to kind of really believe that really we started believing ourselves that it really is some equipotent stochastic population rather than some selective dynamics. Right? So that seemed to be the best explanation of the data so far, right? But that poses sort of, so where are we on this? We have 2 question marks. So the first one says this: well, this diversity is not very different from this diversity, right? They kind of show up in the same way. So when early tumor grows and after relapse looks kind of the same. It doesn't matter what drug you give in some ways. So that tilted the focus to really trying to figure out what this is, right? If you want to now control, again, remember, the question was, we wanted to control relapse by finding out some good genotypes we could target. Now that hypothesis is gone. So then the second idea was, well, let's target these guys because they seem to be a phenotypic state now, right? So the question goes a little bit away from the genotype to the phenotype, right? As you pointed out, right? So we wanted to really phenotype this state now, right? And we have lots of tools now. So what's this? So this is called, drug tolerant persister state in the field, right? And this word persist, I think, was borrowed from bacterial literature, but I don't know. Again, it's always hard to know the sequence of events. So what's the nature of this persistent state is what we wanted to do. And again, now you can sort of get into your molecular genetics, grow these things in a dish, different treatments. These are different controls, and just go do RNA-seq. Right? We didn't do single cell RNA-seq. We were just interested in average property of this residual cells, right? And you can find, these are now sort of very, we do this every day, these kind of things, just sort of PC plot, if you will. And you can see that, so what are we seeing? So this is the DTP state. The regrowth state is actually similar to the control. So this is the control, this is the regrowth. They kind of all go together. But on the same plot, what Sumi decided to plot was actually look at, so this was her idea, which said, look, these cells are kind of paused, right? So they did a lot of staining in terms of looking at the biology of them, and they look like very, very slowly dividing cells. They were not completely paused, but they were very slow. And so their idea was that, this is a phenomena which apparently happens when you take stem cells and starve them. They just pause. They don't die, they wait. And this also happens in the dowers. If you take C. elegans and give it no food, it becomes this dower state. So that dower state and these are all kind of the same. So those guys had this idea, well, let's just look at what these guys are doing, right? And well, it doesn't look that impressive, but this species still separates out these two from those. So they said, well, look, let's just plot this embryonic POSIX signature for these different kind of tumors and see how it stacks up. Okay? So this is random, just to say that you can take DTP state, regrowth, or resistant, right? So remember, the resistant are the ones which have sort of grown after you haven't taken the drug out. So these guys are actually resistant to the drug now. Right? This was the regrowth in a relapse, and this was the DTP state. Okay? Those are the different things. And you find that it's only the DTP that has a high sort of correlation with the embryonic pausing signature. All the other ones are basically after stuff has grown. Right? So the phenotypic state of cells, no matter whether they're resistant, they never saw the drug, or they relapsed, all of them are very similar in their phenotype. Not surprising, they're just growing cells, right? But these guys are actually much more like this paused state. Good? Makes sense? Then they said, well, we're going to push it harder, and said, look, let's just look at leukemias, prostate cancer, melanoma for the MRD. So they had samples of MRD tumors and their gene expression, and they started plotting this embryonic pausing signature across multiple tumors. And these are the tumors which were basically, these were the patients which didn't do very well, where relapse happened. Right? So they basically look at the MRDs, and you see that compared to the parent tumor is much higher signature of this pausing. It's kind of giving you something now, right? But what do you do next? If you start really believing it, well, you do these Kaplan-Meier plots, right? So this is the Kaplan-Meier plot of signature low and high. Right, these are the numbers of patients, so reasonable statistics, 700, 600, 700, 650, 550 patients. And this is, again, in doing this, it turns out that the RNA expression actually gives you another signature to look at, autophagy. So when autophagy is high, basically these guys can't quite make it, right? So they need autophagy in some ways to survive this MRD state. So autophagy is kind of, high. Yeah. Not study. This is just data which was there. This is human. Yeah, it's human data. Yes. Sorry, the question was whether this is a clinical study coming from humans. So yeah, this data is coming from humans. So they're like taking, so of course this was in a dish, right? Up till here was a dish. This was actual human data, right? And then this was also human data, right? This was, I think, for colorectal cancer. The other ones were other tumors. So these were other tumors. The primary line we were studying was from colorectal tumors. So I think this one was colorectal, right?
Were all these patients sequenced, like they had RNA-seq for the minimal residual disease?
I'm imagining.
Okay, all right. So those are matched.
Yeah.
Examples.
Yeah.
Okay.
Yeah, I assume it's a TCGA database.
Could be some,
Okay.
yeah. It's
Yeah,
some, yeah.
makes
I don't
sense.
think there were new experiments. done.
Okay.
This was not experiments we did. No. But this makes a prediction now, right? So far so good. I have some magical signature. What do I do with it? Now, what's the prediction? The prediction is there's some phenotypic state this MRD state needs, and if I can disrupt it, I should be able to do something, right? So again, because autophagy was a nice signature, okay, this is a busy plot. Let me just take you through it. Well, let me give you the logic first, and then I'll show you the plot. The logic is, if I just give chemotherapy, it's going to regress. I'm going to take the chemotherapy out, it's going to come back up. Now, what do I need to do? I need to stop somehow these cells to enter into this new state, which is called DTP. If they cannot enter into it, then when I take the drug off, nothing is going to come back up, right? Because that being in that state helps them survive the chemotherapy. But that's the idea right now, right? So what you can do is you can basically muck with the autophagy pathway, which they require to get into this state. So if you give both drugs, you don't get relapse. If you give any one of them, you get relapse, right? Okay, so this is a harder plot to read a little bit. So this is just DMSO. Somewhere here is when the drug is stopped. So this is CPT11. So if you keep the drug on, it doesn't grow. You take the drug off, the tumor relapses. This is what we saw, right? It's the classic relapse. This SBI is the drug which affects autophagy. It doesn't have a big effect. It really does something, but they grow. Now this green lines is what I want to, which is sort of buried in the bottom, is basically if you give both drugs, even when you take the drugs off, there's no relapse, right? So basically what you've done is hindered the cells to go into this new state, right? That's what the interpretation is, right? Shows something more is going on.
Probably this is coming up on the next two slides, but did you sequence those cells in the green bottom line?
Oh, to just look at genotypes?
To just look at the color, yeah, to look at the distribution.
No.
Okay.
Because this was now phenotypic study.
Sure.
So we were not even tracking barcodes.
Because it would be interesting, I think,
Yeah.
right? Because then you can potentially separate out the entry and the, like
Exit.
the theta and the
Oh, for
ramped
sure.
up.
Yeah, you could get those parameters.
Yeah.
You could get those parameters. Yeah, if you were happy with the ratio. Absolutely. But so this was sort of, and thanks to Sumi now she's sort of really going after some clinical studies, and she has a startup now thinking about how to go after, because if this is really something generic, then they can do much more, right? And I don't think as a theorist I have much more to contribute to that, but that sort of is how this story was. Then we worked on, this one is in a dish. Yeah, this is a prediction. So this is all, that's all what they're doing right now. Now, whatever they can get their hands on. Yeah, exactly. But to me, it was satisfying. You make a prediction, it works out somewhere, right? And then if we, yeah. And we sort of worked. So now I was curious about what happens in temporal. I'm not going to talk too much about it, but it looks like, again, the idea was that this crowding is important, right? So we started seeing some glioblastoma data, and you can see that you get different distributions depending on sort of whether the tumor is diffused or localized. So it's sort of just coarse phenomenology of the tumor will tell you what kind of composition the tumor is going to have. And then you can design whatever you want to do in terms of tackling that. So I won't go too much into it. Here you see sort of a strong effect of selection also. I know it's hard to read these plots, but basically these clones are very, very big compared to the rest of them. Right? So there's a huge tail of them, which are small, and then there's a few clones, 2 or 3 clones, which became very big. It's unlike this red one, but of course they're still big clones, but the rest of them are not too far away from them. Right? So you can start seeing these very different things, depending on really the core structure of the, so these are, so these were not xenographed, these were in situ. So this is also mouse, but they were injecting this in the brain, and then they can track. So as the picture suggests, right? And then they were, whether it's diffused or localized, you can see different things. Okay, maybe I'll pause for a minute if there are more questions on this. I jump to the second one. Yeah.
So I think that, so obviously the situation is different, but in both like bacterial evolution and in cancer, people talk about adaptive dynamics, where because essentially the idea is that you during treatment are always selecting for resistant clones, which is true. I get in, of course, if you're applying it all the time. This was just one application of drug, but what are the consequences of the idea that that in absence of the drug, the growth is essentially neutral? I mean, it's not necessarily neutral, but you know what I'm saying? It's basically neutral dynamics as opposed to selective dynamics. What does that mean potentially for the idea of adaptive dynamics based on?
Right. So I think adaptive dynamics is happening, but it looks like that it may not be the only dominant thing. So it looks like there's a lot of phenotypic things going on, and that may not be a bad way to think about relapse in particular. That's what this is saying. Of course, adaptations are going on, but somehow they were not the dominant effect, right? So, but here they were, for example, here they were. This is adaptation, right? Absolutely adaptation, right? And adaptation from the standing variation, actually, not even from new ones. It could be new, we're not tracking genotypes. So it's not that it can't happen, but depends on the structure of the tumor, that which phenomena would dominate, right? That's, I think, is interesting in some ways.
Yeah, thank you. This is just to, I might have missed this and you might have mentioned, but when you look at the barcoding for the relapse, you see that the heterogeneity distribution as a matrix, let's say, is similar. But like if you look at the same tumor, are the clones in the relapse state the bigger one initially, or is like other clones that were
sequence
smaller?
them, right? So what you would like is, no, that's a great question. What you wanted is, well, I have a tumor which grows, does this.
Yes.
If I sequence it here and here.
Or look at the barcodes.
Barcodes.
Okay.
Then you would like that, oh, the bigger clones are represented here in a bigger way. Or not, right? Is there correlation between these two? That's the question?
Yeah, because I would expect maybe if it's kind of asking the question, which is not purely relevant, but it's just interesting in my mind, is like, were these dormant cells already present? And so they were actually the smaller clones, or did they emerge during treatment?
So good. So our hypothesis is, I think they really emerged. So there's some switching of states happening. Because if they're really dormant, those guys are not going to be there at all in this sample.
Okay.
You're going to be very, very outcompeted very quickly. You have to be growing.
Because I guess like in some way, maybe if I think about some tumors that have like regions.
Yeah, I mean, you're right. It
spatial
could be some
heterogeneity for
necrotic core, which
movements.
something is sitting
Yeah,
there.
exactly. So
That's a great question. Yeah. So I'm trying to convince a collaborator who does things in structured things to really grow, yeah, spheroids, and they actually grow things in wrapped tea bags and things like that, so they can make weird 3D structures. So try to image them and see really these kind of questions. So in some ways we are invoking space, but we have done no spatial measurement, right? No, absolutely. So I don't know the answer, but you know, if they get, yeah.
Yeah, I guess it's kind of a comment. This is very fascinating, by the way.
Thank you.
But you've commented many times that it's not selection that is you're seeing here, but I would argue that it is. It's just environmental and spatial selection. It's not what we think of as like drug-induced, selective pressure, but the environment itself is imposing this ability for some clones to grow large and some clones. So it's just sort of the randomness of the interactions with the environment that is causing some clones to grow.
I should
big
take
and
back.
some
I think there's no
clones to
drug,
grow small.
there's no genotype. Looks like there's no genotype effect.
Yes, yeah.
So I wanted sort of good.
But-
The story I want to talk is
but-
genotype phenotype.
yeah,
Yes,
but
maybe
presumably-
the word is too strong. Yes,
Yeah,
I agree.
and they didn't look at like they just barcoded and injected all the human
Yes.
sample, right?
Yes.
So it's not like they've looked at the particular phenotypes of each of those cells to see if some had larger growth rates or smaller growth rates.
Nothing.
None of this is known.
None of this.
Thank you.
Yeah. So there's some
I have
follow-up
one-
experiments in
one
mice
last
we're doing.
question Before you move,
Yeah.
just to the drug, besides chemotherapy drugs,
Yeah.
did you guys try anything else? Okay, yeah, because chemo kills
Yeah, no,
everything, right?
I'm
And so,
with you.
okay,
Yeah, yeah, chemo, it could be specific to chemo.
right.
I mean, yeah, I don't think I should say anything more. I don't know enough. I could say, but I don't think I should. Good. So, okay, I don't have I have 5 minutes, I guess.
Yeah, I mean, we have,
5 times. Okay, I'll try to, the other part is more theoretical. Yeah, no, it's great. Hey, this is part of it. So the second part is more theoretical and it was done with a student, Matt Smart, who's a postdoc now in Flatiron, and my dear colleague, Anton. So the question here we wanted to ask is, what is happening in this early part, right? So epigenetic switching is an idea that, cells can, again, we wanted to figure out what the role of epigenetics is. But at the same time, there's this idea that microenvironments can feed back, right? So we wanted to just put these two things together in a simple model and see if the phenomenology of how we think about tumor initiation changes, right? So we made the, what Matt likes to call an XYZ model. So we have 3 cell types which have growth rates ABC, and so of course these kind of models have been studied forever, right? What have not traditionally been done in a good way, I think, is that you have these back and forth between cell types. So this is your epigenetic, right? This is some kind of pre-cancer type state, if you will, which is going back and forth, right? This is a mutation. This is a bona fide mutation into a new one, and maybe a second mutation is needed when the tumor really initiates, right? So that's the simple idea. And we wanted to see first, what does this epigenetic do? But the second is there's a feedback, right? So the way we think about feedback is that the rate of this switching really changes as the local environment has more and more tumor cells. The idea being these tumor cells now drive this rate forward. So as you get more cancer cells, they're pushing these X's to turn into Y's more and more, right? So basically you're saying, well, as the tumor grows, it's sort of recruiting more. Does that make sense? It's a positive feedback with epigenetics. This is just saying how the rates are changing as Z changes. So Z is a fraction between 0 and 1, and as Z increases, basically my forward rate goes up and my backward rate falls down. Good? I mean, you can parameterize it in many ways. So these are the simplest equations you could write down, the growth rates. These are the transition rates. We wanted to keep the population size fixed, so we just subtract the mean fitness. So if you add all these 3 without these terms, you'll get some residual growth rate. That's what we are subtracting out of it. This is just the mean growth rate, right? So just keep the population size constant. And okay, this is again that. So a visual of it is this. So you have these green healthy cells, so-called, yellow, these sort of precancerous cells, and they're going back and forth. It's a very slow leak into the yellow. Not much is happening, but once in a while you do. But what happens to these yellow ones are the ones that can mutate. So you start getting a few of these red cells. What these red cells do is change these rates. So they're making this sort of well deeper, if you will, right? Or the barrier lower. So they're trying to increase this rate more and more and reduce this rate. Right? So you can basically start seeing that as Z grows, it kind of actually pulls the whole thing down. It's not just waiting for the mutations, it's actually allowing the epigenetics to kind of drive it even stronger. Right? So we just wanted to see the effect of these two. Okay, so this is in the simple ABC model, without any feedback, if you don't have any feedback from the tumors, no feedback, no microenvironment feedback, right? So there's no pulling down from this tumor. You see these kind of two phases. There's a low Z state, both X and Y are hanging out at some low level, right? Or you can have a second state where Z is the fixed state and there's no other fixed state in the system, right? And you can imagine this happen when the growth of C is pretty high, right? So even if you get a little bit of Z, it just blows up and takes over, right? So this becomes the steady state. And so these are the two steady states that happen in this. You invoke feedback and the phase diagram changes. You get two new phases, and those are what we're going to study a little bit more. And what is happening in these two new phases is this phase is bistable. It's not surprising. You put a positive feedback, you should get some bistability somewhere. So part of this phase, which was low Z, now becomes bistable. Right? So in 4, you basically still have this low state. This low state is still hanging out. Right? This is the part of the low Z state, right? Still there, but you actually create two new states. They come in pairs, unstable and stable, and the new stable one is at Z equals 1. Right? So because it's a positive feedback, it can kind of stabilize that. Good. Once there's a lot of Z, it just pulls everything in that direction. And this is the separatrix. But interestingly, there's another state that opens up in this part, which is in the high Z state, which actually destabilizes this high Z and pulls this down, it pulls this steady state down into the middle. This is basically, you're supposed to be high Z, but now you actually are not able to maintain high Z because remember, this is where B is pretty high, right? And Z was sort of pulling B in particular, right? It was increasing B's. So, wow, the B's take over and the Y goes up and the Z state becomes unstable, right? Good. So intuitively, at least, you can do your bifurcation diagrams and all that for this. This is another way of plotting. So gamma is the strength of this feedback, and mu is the mutation rate, and you can see there's a huge bistable region. So it's plotted in a way to show that. And we wanted to work in this sort of low mu regime. Different things happen at high mu, because then you're just not waiting for anything to happen. You can just already mutate, doesn't matter. So low mutation rates and strong feedback, if you will. So what I wanted to first talk about is the bistable region. Right? So what would happen in this? So again, this is happening without feedback, right? You have this was, remember, the low Z state without feedback, right? So you have a steady state here. This is unstable. Now you both two stable states. If now you look at the transition, so that what you're waiting for is the second mutant from Z, right? So you can ask, how long does it take? What's the first passage time problem, right? Mean first passage time type problem. That's in quantity you can calculate, right? Which is, when did you see on average the first time point when you saw a cell with 2 mutations? Right? What's the fate of it? We are not looking at, but when do you see it first? That's the quantity. So what happens here is you basically come and stabilize here quickly, and your numbers are fluctuating around here because your total population is small. You only have 100 cells, so they're fluctuating around, right? Low populations fluctuate around more, the variance goes up, and you can see basically transition into this Z2 state from a lot of cells, but around in this region. Good. In the other one, what happens is it actually waits up to going to Z. So this is a good way to look at it. So this is the value of Z at which transition happened, and this is the time it took for those transition to happen. Right? So this is the time at which you get a Z2, and this was the value of Z when it happens. So you can see that all of that happens really when Z is pretty low. Right? So you're still sticking around here. Just fluctuating around here, you got a little bit of a Z. The rate was some rate mu of getting it. Right? So you got it. Here what happens is actually jumps over the barrier and reaches the other side. It's fast enough to reach there that most of the transitions are happening there. So what bistability does, it basically makes the chances of getting a second mutation higher. Right? The time it takes to reach Z2 is lower. Not surprising, right? So you can kind of summarize it this way. At large populations, it doesn't matter. Right? It doesn't. The differences are kind of small. At low populations is when things become interesting, where basically having a positive feedback reduces your chance of getting the first thing by a factor of 10, for example. Right? So microenvironment feedback can have a big effect. Right? So that's not surprising. Positive feedback, things should happen faster. And you can calculate these things, anybody's enjoy. So in some ways, this is the other language people use in this is stochastic tunneling versus sequential fixation. If those are interesting, we can talk more. So in some ways, in this part, you're getting sort of some kind of sequential fixation, and here you're seeing stochastic tunneling. Right? So what was surprising is 3. So let me go to 3. So this state 3, which earlier was basically high Z, right? So you can see where I'm going with this. So this was without feedback. So you immediately go to this high Z, and you can get a Z2, right? But now this state is pulled down. So what happens then? So if you look at transitions, here what is happening is, well, it quickly reaches near Z equals 1, and then the transition happens. Here it can't make it to Z equal 1. It's kind of stuck in the middle ground. So what happens is in this case, actually it takes longer to get Z2. So a positive feedback from microenvironment can actually slow it down. Right? That we thought was a bit surprising. And you can then calculate and simulate and whatnot. So here yellow is with feedback. So it takes longer to do it at small population sizes of 100 cells, things like that. Right? So we're thinking really early tumors. Right? Like it could be happening in a crypt, for example, of 100 cells. You're starting this colon thing, and this is all happening at some small scale. Okay? So this was a bit surprising for us, and we had some fun calculating all these things. So you can do all kind of asymptotics and whatnot. Yeah.
I guess this is also relevant for pediatric cancers, like pediatric blood cancers, right?
Yeah.
Where you have this population of stem cells that migrates into the bone marrow from, you originally you have stem cells that are generated in the liver that then transform into the bone marrow that then, so
I did not know that. Okay.
Yeah, that's like in the development of the fetus.
Okay.
So then you could have this variation that crops up and is there pre-existing at birth in some sense, right?
No, absolutely, absolutely. So in some ways, we just wanted to see what this, so the idea is that the feedback should make things faster. That's not true, right? It kind of depends on where it is. And I think Matt looked at some data where he was seeing some puzzling observations with feedback and things like that. I'm not going to take too long. So yeah, okay, just to summarize, so basically in this part, epigenetic precancerous cells may kind of hold the key to tumor progression. Positive feedback can both enhance and slow down, right? So this has a big effect. Having a precancerous thing has a big effect. It's not a small effect. If you're just relying on mutations to make precancerous, it's going to take much longer. This kind of tells you that the rates can really change drastically. This part was talking about, well, phenotypes are important. We don't have to just rely on the genotypic view of cancer. It seems like it could be a dominant effect, really depending on the kind of tumor. And in our hand, it looks like in our way of thinking about it, really look at the gross. It's not even just very specific thing about the tumor, whether it's dense or more dilute, or how is it structured. Those kind of things may really change which phenomenology might be more important, whether you want to do genetics or a phenotype. And this kind of opens up new therapies. If you have a signature you can go after, you can do many things, and that's what Sumi and Catherine have been trying to do. Sorry, okay, I'm going to end. So yeah, I don't have time to talk about this anyways, but so we wanted to really think about now temporal clonal data, to think about trying to make these interactions and really think about subclonal stuff. And the stuff, Trevor's talk yesterday gives you an in into what kind of things we want to try, really to really understand. And the goal here is to really optimize therapy, right? In the sense, if therapy is going to kill the red cell, and if I know, let me see, there's a better one here. I don't know, blue one, right? So if it kills which one, it can tell you, if you knew the interactions, which other subclone might rise, right? So you can kind of predict what might happen depending what kind of therapy you give. So that's sort of is the idea with temporal. If you don't have temporal data, you can't do much. But yeah, I'll be excited to talk to people about this. That's it. It's a very old picture, but it had all the people. It's Matt, Anton, Sophie. Sumi was not there, but yeah, she's. Yeah. Okay, great. Thank you so much.